This is chapter 14 of Campbell's 7th edition biology on genetics, Mendel, and the gene idea. So first off, how do we have all this knowledge? Where do we, kind of the experiments that came from? Well, originally it was thought that genetics was driven by a blending hypothesis where two parents contributed some things and those two things blended together to make the subject that we're talking about. So blue and yellow paints mixed together to make green. That's the blending hypothesis. And that is not necessarily inaccurate now. We know today there are some cases of genetics we'll see later with incomplete dominance where that is sort of the case. The other one was the particulate hypothesis, which talked about inheriting particles or things, units here in this case, which they called genes. So Mendel was the one that accurately documented the particulate hypothesis using garden peas. And what he did was he bred peas very carefully with a brush and chose which peas to cross based on certain characteristics and kept very detailed records of what he saw specifically with the peas. He chose peas because there's lots of different phenotypes that he could test, and also because he could control what mated with what. So here you see an example crossing a purple pea flower with a white pea flower, and he could cut off the stamen of one and the ovule of one, and the stigma and the style. Those are things you don't need to know, of course, but he could control what crossed using the brush. He transferred the pollen, and then you see the offspring that resulted. He would plant them and see what would happen, and he knows, oh, look, they're all purple. So just some info for you. A character is basically the phenotypic trait, like the flower color, and the trait itself is the purple or white flowers. So the actual heritable feature, the character, is the flower color, the big picture, the big idea, and then the trait is the more specific form of the character, in this case purple or white of that character. So Mendel tracked things that he thought were inherited in an either-or manner. You either get this or this. There was no gray area in between. And he also, smartly so, chose to use true breeding characteristics, which for you today, you may be more familiar with the term purebred or pure breeding, where if you cross a poodle with a poodle, what are you going to get? You're going to get a poodle. So that's true breeding. He liked to often take two contrasting true breeding varieties, like purple flower and white flower, and he called this hybridization, and this is the basis for what we call today monohybrid, as in one gene being crossed, or dihybrid, and so on. And the original generation that he crossed, he called the P generation for parent, and then subsequently from that there's the F1, the F2, and so on. So Mendel, initially, the experiment we just briefly talked about before, when he crossed white and purple flowers that were true breeding, he saw that all the offspring were white. He saw that all the offspring were purple, and he said, "What the hell?" No, he probably actually didn't say "what the hell." Mendel was a monk. I hope he didn't say that, but he was curious as to why. So then he took the offspring and he crossed the offspring together. In plants, that's not such a big deal. In humans, that's probably an issue if you're crossing brother and sister. But he saw that a lot of the plants had purple, but then the white flowers reappeared, and this got him wondering. Here you see the white flower reappearing. And he saw that the ratio was in about a 3-to-1 ratio, approximately. It's rarely perfect. It's rarely exactly 3-to-1 in real genetics, but it's pretty close. So he said that the F1 plants, the purple flower factor was affecting the flower color, and that purple flower was dominant and white flower was recessive. He was the first one to consider dominance and recessiveness. And he crossed a lot of different varieties. Here you see all the different dominant traits. Yes, peas can be yellow, but they're generally sold to you as green. Peas can also be wrinkly, but we don't generally like wrinkly peas. We think there's something wrong with them, so it's been bred out. Round is dominant. Lots of different characteristics. So Mendel's model, he had 4 minor ideas that all tried to fit and explain this 3-to-1 inheritance pattern. So the first are that there's different variations of the same gene, and he called these variations alleles. So here you see a homologous pair of chromosomes. Again, this is a pair. One came from the mother, one came from the father, so to speak. I guess I don't know if you can call it that in plants, but we'll call it that anyways. And here you see at the same locus stands for location, on the chromosomes we have an allele for purple flowers and an allele for white flowers. They're both genes for flower color, but there's slight variance on that. You also notice that we're not looking at the X shape of a chromosome. You'll hear me use at times the word chromatid instead of chromosome. Chromatid is just one leg of a chromosome, but keep in mind, however, that the two legs of a chromosome, each one is a chromatid. You put two chromatids together and you get one chromosome, and those two sister chromatids are identical. Here we're only showing you one chromatid and one chromatid of a homologous pair. The second thing that he came up with was that every organism has two alleles for one trait. The genetic locus is represented twice, and thus the homologous pair. Third, if the alleles differ, then one is dominant and affects the organism's appearance, and the other one is recessive, and you can't see it. Fourth was Mendel's law of segregation, where he said that two alleles for a character would end up in different gametes, so that if you have two alleles, in our case we talk, let's say, big A and little a, one gamete will get the big A and one gamete will get the little a. And by gamete we mean egg or sperm. So then we have the F2 generation, and all these mechanisms that Mendel proposed and saw were helpful in explaining this F2 reappearance of the white flower, and you can see that here. Here we have the P generation. You cross them, you get all purple flowers. And these purple flowers, although he didn't really know initially, were heterozygous, big P, little p. And when you cross two heterozygous purple flowers, 1/3 of the, 1 out of 4, or 1/4 of the time, a 3-to-1 ratio, we saw white flowers, little p, little p. Homozygous means that an organism has the same allele at that genetic locus. So on each of the homologous pair, it would be purple and purple, or big P and big P, for example. That's homozygous. And heterozygous is differing alleles at that genetic locus. Phenotype is physical appearance. Genotype is genetic makeup. Here you see examples of that. Notice heterozygous, big P, little p, and homozygous, big P, both have the same phenotype. So in pea plants, genotype is not always obvious, but phenotype is. So sometimes if you're trying to determine the genotype, you can do a test cross. And a test cross is when you take an organism with an unknown genotype, but it's dominant, so you don't know if they're homozygous dominant or heterozygous, and you cross that individual with a homozygous recessive organism who you know genetically is homozygous recessive, and that will help you to determine what the parent, dominant phenotype was that was unknown. Here's a diagram showing you as such. You cross it with a homozygous recessive, and depending on what the offspring ratios are, you can determine if this unknown parent was big P, or big P, little p. And we'll take a pause on this.